All right, page 4, the diversity of cells. Most cells are so small that they can't be seen by the naked eye. So how do scientists find cells? By accident, that's how. The first person to see cells was not even looking for them. All living things are made of tiny structures called cells. A cell is the smallest unit that can perform all the processes necessary for life. Because of their size, cells weren't discovered until the microscopes were invented in the mid 1600s. Cells and the cell theory. Robert Hooke was the first person to describe cells. In 1665, he built a microscope to look at tiny objects. One day, he looked at a thin slice of cork. Cork is found in the bark of cork trees. The cork looked exactly like it was made of little boxes. Hooke named these boxes cells, which meant little rooms in Latin. Hooke's cells were really the outer layers of dead cork cells. Hooke's microscope and his drawing of the cork cells are shown in figure 1. Hooke also looked at the thin slices of living plants. He saw that they too were made of cells. Some cells were even filled with juice. The juicy cells were living cells. Hooke also looked at feathers, fish scales, and the eyes of houseflies, but he spent most of his time looking at plants and fungi. The cells of plants and fungi have cell walls. This makes them really easy to see. Animal cells do not have cell walls. The absence of cell walls makes it harder to see the outline of animal cells. Because Hooke couldn't see their cells, he thought that animals were not made of cells. Finding cells in other organisms. In 1673, Anton van Leeuwenhoek, a Dutch merchant, made his own microscopes. Leeuwenhoek used one of his microscopes to look at pond scum. Leeuwenhoek saw small organisms in the water. He named these organisms animalcules, which means little animals. Today we call these single-celled organisms protists. Leeuwenhoek also looked at animal blood. He saw differences in blood cells from different kinds of animals. For example, blood cells in fish, birds, and frogs are oval. Blood cells in humans and dogs are round and flat. Leeuwenhoek was also the first person to see bacteria, and he discovered that yeast that makes bread dough rise are single-celled organisms. Examples of these types of cells Leeuwenhoek examined are shown in figure 2. The cell theory. Almost 200 years passed before scientists concluded that cells are present in all living things. Scientist Matthias Schleiden studied plants. In 1838, he concluded that all plant parts were made of cells. Theodore Schwann studied animals. In 1839, Schwann concluded that all animal tissues were made of cells. Soon after that, Schwann wrote the first 2 parts of what is now known as the cell theory. It has 3 parts to it. The first one says all organisms are made of 1 or more cells. The second part says the cell is the basic unit of all living things. Then it says later in 1858, Rudolf Virchow, a doctor, stated that all cells could form only from other cells. Virchow also then added the third part of the cell theory, which says all cells come from existing cells. Page 6, cell size. Most cells are too small to be seen without a microscope. It would take 50 human cells to cover the dot on this letter I. A few large cells. Most cells are small. A few, however, are big. The yolk of a chicken egg, shown in figure 3, is one big cell. The egg can be this large because it does not have to take in more nutrients. Many small cells. There is a physical reason why most cells are so small. Cells take in food and get rid of waste through their outer surface. As a cell gets larger, it needs more food and produces more waste. Therefore, more materials pass through its outer surface. As the cell's volume increases, its surface area grows too. But the cell's volume grows faster than its surface area. If a cell gets too large, the cell's surface area will not be large enough to take in enough nutrients or pump out enough waste. So the area of the cell's surface, compared with the cell's volume, limits the cell's size. The ratio of the cell's outer surface area to the cell's volume is called the surface area to volume ratio, which can be calculated using the following equation. Surface area to volume ratio equals the surface area divided by volume.